Okay, I think that was most people in the waiting room for now. So I'm just going to start. Welcome everyone to another installment of Paleo Perks. I hope you're having a great Tuesday, whether it's morning, evening, or midday for you. So today we're going to listen to a talk by Niklas Hohmann from the University of Utrecht in the Netherlands, who is going to talk about recovering trait evolution from incomplete carbonate successions. So the format of today's seminar, if you're new here, we will start with a short welcome and announcements, which is what I'm doing now. Then we'll have Niklas' talk, which will be around 30 minutes. This will be followed by around 10 minutes of a moderated Q&A. And Niklas has also agreed to stay for tea time. So if you want to have an informal chat afterwards, then you are free to stay. And please don't forget to send your questions via the chat to the questions at PaleoPerks host, which is Ohana today. You can start sending your questions during the talk or during the Q&A itself. A little bit more housekeeping, especially if you're new here. PaleoPerks values the participation of all folks interested in the paleo sciences. And please remember to abide by our code of conduct during today's seminar. If you have received this link, then you should have read our code of conduct and agreed to it. Please also mute yourself for the duration of the talk out of respect to the speaker. You should not be able to unmute yourself, but if you do find yourself unmuted, please mute yourself again. Again, you can ask questions by chatting to the questions at PaleoPerks host, or you can also use the raise hand function during the Q&A. Then you can ask it yourself. If you have any technical questions or issues, you can also send them to the questions at PaleoPerks host. Then we also have closed captions built into Zoom. You can use the CC button to either show or hide them. And finally, we're also always looking for new speakers. So if you know an outstanding ECR or you want to give a talk yourself, you're also very free to self-nominate. Please follow the link here. It will also be dropped in the chat shortly. And we always appreciate any feedback or demographic information, which is completely anonymous. So if you want to fill that out, a link to this feedback form will also be dropped in the chat. So that was all the housekeeping stuff. Then I would like to introduce today's speaker, Niklas Hohmann. So Niklas did his bachelor at the University of Erlangen in Nuremberg in Germany. And he did his bachelor's in mathematics and geoscience, followed by a master's at the same university in paleobiology. And now he's doing his PhD at the Utrecht University in the Netherlands. And what that is about is what he is going to tell you about, hopefully today. So Niklas, feel free to start your talk and share the screen, and I look forward to it.
All right, everyone. Yeah, thanks for attending. It's a pleasure to be invited here. And I will be talking about recovering trait evolution from incomplete carbonate successions. And as a paleontologist, for me, it is like almost intuitive. Like it's very intuitive that fossils are something amazing, right? We can use fossils to trace evolution over timescales that are not accessible to direct human experimentation or observation. And they really open the opportunity to observe evolution over millions and millions of years, which is something which I still find extraordinary. But not all evolutionary biologists actually agree with this. And I want to open my talk with a quote from Charles Darwin himself, who was very, very deeply skeptical of the usage of fossil data. This is a quote taken directly from The Origin of Species. And he says, "Geology assuredly does not reveal any such finely graduated organic chain. And this perhaps is the most obvious and gravest objection which can be urged against my theory of evolution. The explanation lies, as I believe, in the extreme imperfection of the geological record." So Darwin himself was deeply, deeply skeptical of the usage of fossil data, mainly because he considered the fossil record to be extremely incomplete and just be an extremely unreliable source of information. And that is something that is actually still around today. So when you go to a evolutionary biology conference, a lot of like neontologists will be deeply skeptical when you use data from fossils to infer evolutionary change. The good thing is that Darwin has actually been dead for a long time, and we know much, much more about geology than he did and geologists of his time did. So the question I want to address today is, can we actually, using a modern understanding of geology and geological incompleteness, can we assess how does incompleteness affect the recovery of evolutionary history, to kind of either support or contradict Darwin's claim that incompleteness is a hindrance to the recovery of evolutionary change. Because this is a very, very broad question, I'm going to narrow it down. And I'm going to specifically focus on one type of incompleteness, which is stratigraphic incompleteness, which is essentially just the ratio of how much time is preserved versus how much time is being destroyed, either by erosion or by non-deposition in the first place. So that is usually a number between 0 and 1. To give you an estimate, empirical estimates of incompleteness are somewhere between 3 and of completeness are between 3 and 30%. That means more than 70% of evolutionary history is destroyed in the first place before even being recorded. And because evolution is also a very, very broad umbrella term, I'm specifically going to focus on trait evolution within one lineage here, but you will see that a lot of the arguments I'm going to provide are actually immediately abstracting into other types of evolution. I'm going to specifically focus on one depositional system, which are tropical carbonate platforms. In this case, this is a satellite picture of the Bahama Banks. And the argument why carbonate platforms is rather straightforward. They form a large portion of the fossil record, so they are highly relevant. We know they have a high diversity and also a high competition. So we know that we expect that if there is something interesting evolutionary happening, we expect it also to be recorded within a carbonate platform. They're also very interesting from a sedimentological perspective, because in contrast to siliciclastic systems, they are actually built by organisms themselves. So siliciclastic systems are kind of dead. They're just sand and mud transported from a point source into the basin. Whereas a carbonate platform is kind of like a breathing, living organism that responds to external forcing, such as changes in insulation or changes in sea level. And that makes it very, very interesting because it can give rise to a lot of interesting interactions. And also carbonate platforms have kind of been understudied in stratigraphic paleobiology. So a lot of the classic studies in stratigraphic paleobiology specifically focus on siliciclastic systems, which are rather well understood now, or there has been a recent push more towards terrestrial systems, but there is really nobody been working on carbonate systems so far. For this specific study, we used two stratigraphic scenarios, and they're based on simulated carbonate platforms. So here, these are scenarios A and B. And this is a computer simulation of a carbonate platform. On the left side is onshore, on the right side is the deep sea. And then you can see the different depositional environments across the platform, from the lagoons, the back reef, the forereef with the high wave energy, and then essentially going into the slope where we have continuous shedding of sediment from the carbonate platform into the slope. Something that is very interesting about these platforms is really how distinct their geometry. So we have this flat platform top and then a drastic sharp angle, and then we transition into the slope. We simulate two different scenarios here, and they mainly differ by the external drivers. So they both have the same simulation settings except the sea level. Scenario A is based on a very, very slow sea level change based on a sinusoidal curve, whereas for scenario B, we have a much, much faster sea level change based on the last 2.5 million years based on the Miller et al. Sea level curve. And the figures essentially show you a transect through the whole platform, and the different colors correspond to different lithophases. I will say a little bit more about the simulation that goes into these platforms later on. Then here are the chronostratigraphic diagrams of these two platforms. On the right, you can see the sea level curve. Again, here you have this slow sinusoidal change with some higher order cyclicity there. And for the platform B, we have the rapid changes in sea level based on the Miller et al. Sea level curve. The chronostratigraphic diagrams are a bit complex to read, so I'm going to run you through it. Essentially, everything is white, is non-deposition. So there's no record in everything that is white here. You can read them from the bottom to the top. So if we look at 2 kilometers from shore, if there is a color, this is deposition of the corresponding facies. and then white means there's a gap. We have short gap, deposition, short gap, a little bit of deposition, and then a long gap where there's almost 500,000 years of no deposition at all. And here you already see there's, depending on the driver of the carbonate platform growth, the geometry of the gaps is very different. So in scenario A, we have these huge gaps that span the whole platform. So if there is erosion, there's erosion on the whole platform. In scenario B, we also see a erosion over the whole platform top, but the gaps are much more frequent and much shorter. So that is something you should keep in mind. For scenario A, we have a slow sea level change and a few but very long gaps, whereas in scenario B, there's fast sea level change resulting in many very short gaps. And based on these stratigraphic scenarios, we can extract what is called an H-depth model. And they are essentially, you can think of them as a coordinate transformation between the time domain and the stratigraphic domain. So at the x-axis, you see time in the simulation time, and on the y-axis is stratigraphic height. And let's just take the 8 kilometer H-depth model. This essentially tells you that at this point in simulation, this stratigraphic position was deposited. And you can already guess that there's a lot of stratigraphic information contained in these H-depth models. For example, the sedimentation rate is actually the slope of the H-depth model. And these gaps here are all times of erosion. And now you can put a better intuition onto these H-depth models and also the difference between scenario A and B. Here in these H-depth models, you clearly see long gaps in scenario A that are associated with drops in sea level. And these gaps are actually throughout all of the H-depth models. So independent of where you look on the platform, anywhere on the platform top, there's erosions during time of sea level drop. And scenario B is very different. You see, there's a lot of short gaps interrupted by short bursts of sedimentation. And the discrepancy between close to shore and further offshore is not that drastic. And now comes the interesting part, because we have this is the stratigraphic foundation. But because we know what time is deposited where, we can actually combine that with simulations of evolution. And for this study, we only looked at 3 very simple modes of evolution. They are discussed in the literature. This is stasis, unbiased random walk, and biased random walk. They are not necessarily the most empirically plausible modes of evolution, but think of them as useful end members that we can use to study the effects of stratigraphic biases. So stasis is kind of self-explanatory. It's just the trait really doesn't change with time. There's a mean value here, and there's a little bit noise around it. And for the trait, we just simulated abstract traits. If you want to give it a bit more context, think of these traits, for example, as shape or body size. So stasis would then simply mean that the body size remains the same, and there's just some background noise around the mean body size. Then the next model is the unbiased random walk model. That's a bit more complex. That essentially means that in every time increment, the trait either increases or decreases, and that increase is perfectly random, but symmetric. So the increase is equally likely as a decrease. And depending on the background randomness, you can get these quite large excursions, but overall it is just a noise model. So what's special about these unbiased random walk models, they can produce quite large excursions, but in the end, you always tend to end up in the middle. And then as a third model, which is what we call biased random walk, you can think of this as directional evolution with a little bit of background noise. In this case, this would, for example, correspond to the selection for a larger body size. And now we have the 2 main ingredients in place, which is a model of evolution, but also a model of stratigraphy and stratigraphic biases. And when we combine them, we actually get some really cool results. So this is the first result. So we only looked at one place in the carbonate platform, which is 6 kilometers on shore in scenario A. So remember, this is on the platform top. And then we compared what is the true evolutionary change, which is in the bottom row, versus what is the signal that we actually recover from the carbon platform if we just sample the stratigraphic section that we simulated naively. And for stasis, this is actually pretty straightforward. If you originally have stasis, you recover stasis, which is actually not really surprising, because if nothing exciting happens in the first place and you're removing a part of it because of erosion or hiatuses, still nothing exciting happens, right? And for the random walk model, it gets a bit more interesting because you can actually see here this drastic jump. And that is at around 150 meters stratigraphic height. And if you go back, and this was 6 kilometers from shore at 150 meters, this is exactly the stratigraphic position where we have a large hiatus of 500,000 years. So with stratigraphic context, this jump makes perfect sense, because this is where the hiatus is. We're removing 500,000 years of evolutionary history, and that leads to this drastic jump. But if you do not have that stratigraphic context, suddenly you might observe a taxon that has a constant body size, and then within a meter or two, the body size drastically decreases. So this is really prone to misinterpretation, that if you don't have the stratigraphic context, you might think this is an evolutionary signal and even maybe identify this as a separate taxon. Interestingly, for the random walk, this is not something you observe in all lineages, essentially because they're random in their direction. It only is visible within a few of the simulated lineages. And that effect gets even more drastic for the directional random walk, where we start with a really gradual increase. But when you interpret the changes in traits naively in the stratigraphic domain, you end up with these two huge jumps here. And if you don't recognize these hiatus surfaces, then this looks perfectly like punctuated equilibrium. Relative stasis in trait value, followed by a rapid jump, followed by a relative constancy, followed by a jump. And I think this really highlights that having stratigraphic context in observations of evolution is really crucial. But another thing is also very interesting. So we looked at the same location in the Carbonate platform, but the degree of bias really depends on the original signal. So if the original signal was stasis, we're fine, right? There's no way to misinterpret this. But the more directional we actually get in our true evolutionary signal, the more prone we are to biases. And this is a really, like, what we see throughout that study is that the stratigraphic record really favors the recognition of both stasis, because if you remove something from no change, it is still no change, but also it favors the recognition of these punctuations when you actually accumulate change over these hiatuses. Another thing is, what we also see is a huge spatial heterogeneity in preservation. So this is the same original evolutionary signal. It's the directional random walk again, but this time sampled at different locations in the same Carbonate platform. So this is scenario A. And here again, in 2 kilometers offshore, you see these two drastic jumps that coincide with these long gaps in the record. But 8 kilometers offshore, so the further we move away from the platform top and towards the slope, suddenly the first jump completely disappears, because there's no long hiatus on the slope actually being preserved. So we get a very different evolutionary signal preserved. Again, if you don't have the stratigraphic context for this, you might misinterpret this for having two very different evolutionary dynamics at different parts of the platform, whereas it is actually just a differential preservation at different parts of the platform. And the pattern 12 kilometers offshore on the more on the slope of the platform, it looks quite similar to the 8 kilometers, but the interesting thing is here, this peak is actually not associated with a gap. And that is something I found very eye-opening. So a lot of the discussion about stratigraphic biases is always about hiatuses and erosion, etc. But this peak here is actually due to stratigraphic condensation. So we have a reduced sedimentation rate, which means we have more time preserved in the same thickness of rock, and that leads to these apparent increases in evolutionary rate on the offshore. So not every bias is actually associated with a gap. You really need to understand your local stratigraphic and sedimentological context. And now for the million-dollar question, what is the actual effect of stratigraphic incompleteness? For this, we again took the directional random walk as kind of like a truth and a baseline. And then we looked for two locations in the Carbonate platform that had very similar completeness. So this one is in platform A with a 33% completeness, and see the right side is around 35% completeness. But this time in Carbonate platform B, remember, that's the one with the short but frequent gaps. And although the completeness is extremely similar, the preservation is, very, very different. So in Carbonate platform A, you have these jumps again associated with these long gaps. But in Carbonate platform B, although the completeness is almost identical, it's like almost a perfect preservation of the original true signal. And the main difference between them is that in scenario B, we have a lot of gaps. That is not for debate, but they are much shorter and very, very frequent. And that avoids actually producing these drastic jumps. So the main takeaway from this is that it's not the actual completeness that matters, but rather, do you have long gaps? In this case, because the gaps are so short, it's just essentially reducing the temporal resolution, but not introducing any larger biases. And the cool thing is, because we have these forward models, we can make very, very systematic observations of how the gap duration and the completeness actually changes throughout these scenarios. So this is a crowded picture, so I'm going to run you through it slowly. Black one is the actual stratigraphic completeness. And here you can already see that the completeness is highest around 10 to 12 kilometers from shore. And that is actually on the slope, because the slope is being continuously fed with sediment from the platform top. And then the closer you get to the shore, the lower your completeness actually gets. And then here you also see the dark red line. This is the major takeaway. The maximum hiatus duration in scenario A is much higher than the maximum hiatus duration in scenario B. And this is really what we found drives the dissimilarity between the true evolutionary change and the evolutionary change that we see in the stratigraphic record. Before I wrap this up, I just want to briefly show you some of the software that we develop in our project, because software is very close to our heart, and there are some really cool pieces, and I think this is really interesting for the community. The Carbonate platform model, the Carbonate platform simulation I showed you were run in an old model, which is called Carbonate. We are developing a new in-house model called Carbokitten, which is obviously derived. It's based on Carbonate and it's written in Julia, which is a new scientific computation software. It is incredibly fast. So it's faster by a factor of almost 100, and it is open source. So if you want to interact with it, all the source code is there. The documentation is excellent. I invite you to play with it. The interesting thing about modeling Carbonate platforms is really that they are not purely physical models, but they're also biogenic models, because we specifically model how organisms live and grow and compete for space. So that is kind of like the hardest scientific part in modeling a Carbonate platform versus a traditional siliciclastic system. Then there's also two R packages. One was just recently published in Methods in Ecology and Evolution. They are kind of like the computational backbone of this study that I showed you. ADM Tools does the main age depth modeling. It kind of takes care of all the stratigraphy and the heavy lifting there. And StratPal is for the stratigraphic paleobiology part. So it does take care more of the ecology, taphonomy, and evolutionary biology side. And just to show off this software a little bit, I just want to show you a little example. So this is a plot that I just made 10 minutes before the talk actually started. These are the number of last occurrences, so the last time you find a species versus a stratigraphic position in the simulated Carbonate platform that I showed you. Now the question to the audience is, where is the mass extinction? Is it at 1 or is it at 2? Just type your answers in the chat, please.
Yeah, once again, everyone, I really invite you to put your answers in the chat. So just type a 1 or 2 in the chat to everyone, and I will try to compile the answers. So I give it a minute, and then I'll start counting.
Oh, nice. I see the answers coming in.
So far, if I counted correctly, I see 6 answers for answer 2, several with a question mark, and 3 answers for 1.
Right.
And then 1 answer is cheating, who says in the middle. Or maybe that's correct. I don't know.
Okay, so I'm going to show you the H depth model, which is again from 2 kilometers offshore, I think here. And with 1 is labeled the hiatus that corresponds to the stratigraphic position of the first mass extinction. With 2 is labeled the hiatus that corresponds to the stratigraphic position of the second mass extinction, or the second peak in last occurrences. And it's actually a trick question. There is not a single mass extinction here. What I simulated is a constant rate of last occurrences under this H depth model. The peaks in last occurrences that you see here are simply because all the last occurrences that happened during the first hiatus will actually accumulate on that hiatus surface. So this is an absolute null scenario. No evolutionary change, nothing exciting is happening, but nonetheless, you get these 2 drastic peaks observable in the stratigraphic domain. And just to show you a little bit how the working of these packages is, on the left side, this is the code, the 2 lines to actually construct the H-depth model. And on the right side is all the code to, the first line simulates the last occurrences, the second line introduces the stratigraphic bias, and the third line plots the histogram. So it's really, really tailored to be simple and easy to use. So if you want to play around with this, I invite you to. We have plenty of teaching materials on this. We have a whole workshop that you can either give or work through yourself. It's available on GitHub. And I think for the people among you who also teach, we also have a really, really nice Shiny app, which I just want to show as a last thing here. All right, so this is the Shiny app. We use this commonly to teach our undergrads because our models are quite complex and people tend to get very, very intimidated by code. So we developed this app to let people interact with our models without actually having to enter any code or interact with any type of programming language. I'm going to jump right into the complex thing. And this is essentially an interactive summary of the study I showed you. So on the left side, you can specify what kind of evolutionary scenario you want. So I'm just going to make a slightly directional random walk. And this is what you see here down at the bottom. This is trait evolution in the time domain. This is our evolutionary truth. Then in the middle here, this is the H-depth model with the gaps. And here on the left side, you see the actual stratigraphic expression of this evolutionary change. And then using these sliders, you can actually just explore how the preservation changes along an onshore-offshore gradient. So you get the different hiatuses here, and everything will always be updated live. You can also highlight the gaps to kind of like visually emphasize that, and also emphasize where the hiatus surfaces in your stratigraphic column would be. And then you can nicely see that this jump in trait value actually perfectly coincides with a hiatus surface. And then what's also interesting, you can immediately display the driver of the stratigraphic architecture. So here you see the original sea level change. But then here in the stratigraphic domain C, also the preservation of the sea level change is of course biased. So instead of the slow drop in sea level, you get a rapid drop because all the record of the falling sea level is actually being eroded. And the increase in trait value coincides actually with a drop in sea level, which is, again, if you don't have the stratigraphic context, is rather easy to misinterpret. All right. All right, then to wrap this up, in the project, we develop tools for computational stratigraphic paleobiology. We invite you to interact with these tools. We're always happy to have new contributors, get feedback on them. Yeah, just go to our webpage and play around with it. We found in our study that actually the preservation is not a one-size-fits-all thing. How biased your actual record is strongly depends on your primary signal that you are trying to observe. And then also, it is really not that much about the incompleteness of the record. It is really much more about how long are your actual gaps. You can think of it as if you're like going to the movies and you're missing half of the movie. It is a huge difference if you miss the second half of the movie or if you're missing every second frame. If you miss the second half, there's no way you can understand the plot. But if you're missing every second frame, you still have a good chance to actually follow the plot, although at a slightly lower resolution. And with that, I think the really interesting question to understand what drives the bias of the evolutionary record is to understand what are the external drivers of stratigraphic architectures. And that's it from my side, and I'm happy to take any questions. Thank you.
Thank you so much for that interesting talk, Niklas, and also thanks for the audience for that nice interaction that we had. Yeah, before we start with the questions, I would like to briefly remind everyone that we're always looking for new nominees. So my colleague will briefly drop them in the chat. And then we can start with the first questions. So you can have, you can either ask them anonymously or you can ask them with your name attached. So the first one we have is by Chime, and he asks, carbonate slopes often have mass transport features, including large transitional slides, and the offshore position means that often there is little diagenetic sedimentation. Can you handle that?
So we do not model mass transports explicitly because our model is time averaged. So we usually model on time increments of 1,000 years, simply to be able to run the model long enough, like 2 to 4 million years. So this is not explicitly modeled, but we have sediment transport in the model. Yes, that kind of like is a time averaged version of mass transport downslope.
Okay, thank you. I hope that answers the question. Then we have an anonymous question who asked, do you plan on running the model with evolutionary models more complex than the biased random walk, etc.?
And that is actually pretty easy. So the software packages are designed to work with almost any evolutionary model, because what the software does is it takes a point in time and replaces it by a stratigraphic position or the other way around. So it's actually completely independent of the evolutionary model you plug in the first place. As of now, what I haven't shown, it also works for phylogenetic trees. So whether, like mathematically, it doesn't matter whether you examine preservation of a phylogenetic tree or a lineage. It's identical. So that the state space of the model is absolutely irrelevant, positively.
Thank you. Briefly mentioned that Tom Dines says film analog, that's superb. Thank you very much. Thanks. That was a really helpful analog, I agree. We have a few more anonymous questions. The next one asks, how detailed is the biological component of carbokitten? Can you model, for example, distinct Paleozoic versus Mesozoic reef platforms?
So yeah, as I mentioned, the biological part is the hard part. What we currently do is we have a model that uses what's called a cellular automata to generate spatial heterogeneity, which essentially models carbonate factories that compete for space. So they have a certain dispersal, they have a certain competition, and then they occupy the carbonate platform and grow also in dependency of their neighbors. These factories have, for example, distinct tolerance to neighbors, distinct tolerance to wave energy, or require a certain amount of, for example, sunlight, so insulation. We can vary all these parameters. That is easy. The hard part is actually getting empirically justified parameters. So we can model Paleozoic carbonate factories to the degree in which we can reasonably constrain the parameters, their growth parameters. Yeah, it's a hard problem. And I assume a Paleozoic carbonate platform would look very, very differently. But excellent question.
Thank you. So more input from paleobiologists is always wanted, I assume.
Always. Like constraining parameters is always a good job.
We have a few more questions. And if you have questions, again, please send them to the questions@paleoperks host. Then we will read it out. So this one is about the software. They ask, thank you for your talk and sharing the software. When there are other softwares available with similar functionality for analysis, how would you recommend analyzing the data? Stick to one software or use multiple?
I would say stick with what is established and tested, and then try to plug them together. And the challenging part is really the plugging together. So the package that I developed, the StratPal and ADM tools, it currently interoperates smoothly with PaleoTS for time series analysis and EvoTS for evolutionary time series analysis. I'm currently working on integration with FossilSim and PaleoTree. But if you are not scared of actually wrangling a little bit with objects in R, then it should essentially, you can use any type of analysis. Although we try to make this simpler, but I cannot accommodate all software packages there.
Thank you. And then we have, for now, the final question. But if there's any more questions, please ask us. So this one said, great talk. What would be your top tips for paleontologists that may not be so familiar with analyzing the stratigraphic record, identifying hiatuses, etc.?
My top tip would be, essentially be three things. Get a very experienced collaborator and discuss the hell out of the problem with them. First thing, there is nothing like a good scientific discussion. The second thing is get a forward model and play with it, because it can give you insights into dynamics that are otherwise hard to understand and timescales you usually do not have access to. And then go in the field and double check your forward model with your collaborator in the field. I think using all three really is the way to go forward, because only the model can be very, like, it can be a very academic exercise, but the benchmark is always the field.
Thank you. We got one more question, which I will put it in the chat now. Thank you for a wonderful talk. This is such a cool and important research. A rather broad question. For those of us who rely on methods of sampling standardization, for example, statistical techniques such as coverage-based rarefaction, to estimate paleodiversity change, aside from documenting and caveating the issues that you mentioned in your talk, Is there something that you would highly recommend that we include in our analysis, or a pre-step that we should take?
Oh, that's a big one. That's like a PhD on itself. The thing is, there are a few methods out there that can account, for example, for like facies change over gaps, etc., etc. But they're all extremely complex. So out of the top of my head, I think giving a realistic assessment of the caveats is the way forward. For everything else, I would have to think longer to give like a proper answer to this question. So I cannot answer this in full extent now.
I think that's a very reasonable answer. We received one more question, which says, what type of sedimentation would you say is the most common, the described A and B in the presentation? Or does this change over the geological records? So I guess with lots of long gaps or many short gaps.
It really depends on whether you're like an interglacial or not. So how the sea level responds. For the sake of evolutionary biology, I hope it's scenario B, because then we have actually a much better opportunity to actually recover signals. So something with actually a strong Milankovitch forcing of the sea level would be perfect, because it means we have a lot of gaps, but they're all reasonably short.
Thank you. With that, we have currently reached the end of our questions queue. I will wait a few more seconds, so if there's any questions that you really want to ask, please send them to the questions host now. Or if you want to ask them in a more relaxed setting, you can also wait until tea time, of course. It seems that there's no more questions, but a lot of people are saying that you had a great talk, which I fully agree with. So with that, I would like to end the Q&A part of this session. And before we go into tea time, I will briefly take over the screen to share some PaleoPerks info. Yes, so as usual, thank you all for joining us at this great talk. I really enjoyed it. Again, please fill out the weekly feedback form or nominate a speaker that you think would give a great talk. We hope you to join us next week. The speaker and topic are to be announced. And up next, there is tea time with today's speaker. So this is, again, an informal conversation about the talk in a relaxed setting where you can turn on your video and you can unmute yourself and just have a chat. We always have an icebreaker question of the week, which today is, what is your favorite fossil? So with that, we're having a little break before we all come back. So get up, walk around. If you have any pets, you can bring them with you. And in 2 minutes, I will see those of you that stay for tea time. Those of you that have to move, have to go, I hope to see you next week.